In previous video we have discussed about RB, that is the governor of proliferation. Okay, we are discussing about the cancer genes. In previous videos we have discussed about proto-oncogenes, and now we are with tumor suppressor gene, in which first, that is RB, we have discussed. Now in this video we are going to discuss about TP53. So the first important point about TP53 is that this is known as guardian of genome. Clear? RB was known as governor of proliferation. This is known as guardian of genome. Clear? It is a tumor suppressor gene that regulates the cell cycle progression, DNA repair, cellular senescence, and apoptosis. Okay, and it is the most frequently mutated gene in your human cancer. It is the most frequently mutated gene in your human cancer. Clear? Now, there is loss of function mutation in TP53, which is located on 17p13.1, are found in more than 50% of cancer. Clear? So the loss of function mutation of this gene is found in more than 50% of cancer. Clear? Moreover, TP53 mutation occurs with some frequently in virtually every type of cancer. Okay, some frequency in virtually every type of cancer, including your carcinomas of lung, colon, and breast. Lung, colon, and breast, which is the three leading causes of cancer death. Clear? Now, in most cases, mutations are present in TP53 alleles and are acquired in somatic cells. Clear? Less commonly, individuals inherit one mutated TP53 allele. So most commonly, it is your acquired in somatic cells. Okay, less commonly inherited form. Getting? As in the case of RB tumor suppressor, which we have discussed, and retinoblastoma, inheritance of a mutated copy of TP53 predisposes individuals to malignant tumors because only one hit is required, okay, to abrogate the TP53 function. So it will increase the risk of cancer. Now, such individuals said to have Lee-Frommeni syndrome. This is another MCQ question. Lee-Frommeni syndrome. Clear? So inheritance of a mutated copy of TP53 predisposes individuals to malignant tumors because only one additional hit in the lone normal allele is needed to abrogate the TP53 function. So such individuals are said to have Lee-Frommeni syndrome. Clear? And this syndrome have a 25-fold greater chance of developing a malignant tumor by the age of your 50 than the general population. Clear? So this is important MCQ question, that is Lee-Frommeni syndrome is associated with which gene? So answer will be TP53. Clear? Now, this people with Lee-Frommeni syndrome often develop cancer at young ages and more likely to suffer from multiple primary tumors of various types than are normal individuals. Clear? So this is the basic introduction about TP53. Now, going into detail. So this TP53 encodes the protein P53, which is tightly regulated at several levels. Clear? Your analogous to RB, many tumors lacking TP53 mutations have instead other mutations which are affecting the proteins that regulate P53 functions. Clear? For example, your MDM2 and its related proteins of the MDM2 family stimulate the degradation of your P53. So it stimulated P53 degradation. Okay, MDM2 and MDM2 related proteins stimulate the degradation of P53. Clear? And these proteins are frequently overexpressed in malignancy with normal TP53 alleles. So suppose this, what is the function of MDM2? So MDM2 induces P53 degradation. Clear? And if amount of MDM2 increases, then there will be increased P53 degradation. So P53 degradation will increase. Then there will be increased proliferation signals because P53 is a type of tumor suppressor gene. It is anti-proliferative. Clear? And if P53 degradation increases, means P53 is less inside the cell. Because of more degradation, the amount will be less of P53. Okay, so there will increase proliferation inside the cell. Clear? So the MDM2 gene is amplified in your 33% of human sarcomas. Clear? Leading to a functional deficiency of P53 in these tumors. Clear? So this is a very important point, that is MDM2 and its related proteins of the MDM2 family. Also, like RB, the transforming proteins of several DNA viruses bind P53 and promotes its degradation. The best known of these viral oncoproteins is your E6 protein of human papilloma virus. Okay, E6 protein of human papilloma virus. Clear? And it is also associated with your squamous cell carcinomas of the head and neck. Clear? So best known of these viral oncoproteins is E6 protein of high risk human papilloma virus, which have causative roles in cervical carcinoma and a subset of squamous cell carcinoma of the head and neck. Clear? So this is important MCQ question, that is E6 protein and this MDM2. Now moving to the next, that the frequent loss of P53 mutation in human tumors reflects its critical role in prevention of cancer development. Okay, P53 carries out this role by serving as the focal point of a large network of signals that senses your cellular stress. So P53 senses cellular stress, primarily your DNA damage. Clear? Primarily DNA damage, which also includes your shortened telomeres. Shortened telomeres, your hypoxia condition. Clear? So this type of this type of signals can be sensed by your P53. Clear? So P53 senses your cellular stress, primarily DNA damage, but also shortened telomeres, hypoxia, and stress which is caused by excessive pro-growth signaling. Okay, as may occur in cells bearing mutation in genes such as RAS and MYC. These all are sensed by your these all things are sensed by your P53. Clear? In non-stressed, suppose in what is the function of P53 in non-stressed healthy cells? So in non-stressed healthy cells, P53 is held, okay, with association of MDM2. Okay, P53 is held in association with MDM2, an enzyme that ubiquitinate P53, leading to its degradation by proteasomes. Okay, so MDM2 is responsible for regulating the P53 in your non-stressed and healthy cells. Clear? In a stressed cell, which we have discussed, in a stressed cells, however, P53 is released from inhibitory effect of MDM2 via two major mechanisms, which we are going to discuss. Okay, and those two major mechanisms depends on the nature of stress. Clear? Which type of stress is there? Clear? We have discussed about several stresses, that is DNA damage, short telomeres, hypoxia, the other stresses. So in non-stress healthy cells, the P53 protein is binded to MDM2, and in a stressed cell, the P53 is released freely by two major mechanisms that depends on the nature of the stress it is facing. So the first is your DNA damage and hypoxia. Clear? So the key initiator of the P53 activation following DNA damage or in cells which are exposed to hypoxia are two related protein kinases. Mind it, two related protein kinases, that is ATM and ATR. So ataxia, telangiectasia mutated. Okay, ataxia, telangiectasia and RAD3 related. Clear? As the name implies, the ATM gene was originally identified as germline mutation in individuals with your ataxia telangiectasia. Clear? And persons with this disease, ataxia telangiectasia, okay, which is characterized by inability to repair certain kinds of DNA damage and suffers from an increased incidence of cancer. Clear? Now, the types of damage sensed by ATM and ATR are different, but the downstream effects are similar. Once triggered, both ATM and ATR stimulate the phosphorylation of a number of proteins, including your P53 and MDM2. And this post-translational modification, okay, disrupt the binding and degradation of P53 by MDM2, and this will lead to release of free P53. Clear? There will be no degradation of P53 by MDM2 if there is phosphorylation of P53 and MDM2, which is done by your ATM and ATR genes. Clear? Now the second one is your oncogenic stress. So in this activation of oncoproteins such as your RAS leads to sustained supraphysiological signaling through pro-growth pathway such as your MAP kinase pathway or PI3K AKT pathway. Okay, through unknown mechanism, this aberrant signal creates cellular stresses and lead to increased expression of your P14 ARF. This will lead to increased expression of P14 ARF, which is coded by CDKN2A tumor suppressor gene. Okay, and this P14 ARF will bind to MDM2. This P14 ARF will bind to MDM2 and displaces P53, again allowing high P53 levels inside the cell. Clear? Okay, so this is about how MDM2 releases P53. Clear? So involvement of ATM and ATR genes and expression of P14 and ARF. Now coming to the functions. So once activated, P53 thwarts neoplastic transformation by either transient cell cycle arrest or senescence or programmed cell death apoptosis. Clear? Most of these effects are believed to stem from P53's ability to function as a transcription factor. Clear? P53 binds DNA in a sequence-specific fashion and activates the transcription of hundreds of different target genes with P53 binding elements. Clear? The key target genes that execute the function of P53 are not completely defined but appear to fall in three major categories. The first one, that those causes your cell cycle arrest. The second one is that causes apoptosis. The third one is those that enhance your catabolic metabolism. Okay, that those that enhance catabolic metabolism and inhibit your anabolic metabolism. Clear? Now the latter group of genes make intuitive senses. There is no need for a cell that has stopped its cell cycle progression to continue to synthesize the large amounts of macromolecules that are needed for cell growth and division. Clear? Now also included in the list of P53 target genes are those which are encoding two kinds of regulatory RNA, which you are knowing, that is microRNAs, okay, and long intervening non-coding RNA, that is LINC. Clear? And they presumably help to coordinate the P53 dependent cellular response to stress. Now we will discuss each of these. So first we will discuss transient P53 induced cell cycle arrest. So first we will discuss about cell cycle arrest. So rapid onset P53 mediated cell cycle arrest may be considered as a primordial response to DNA damage. Okay, it occurs in late G1 phase and is caused in part by P53 dependent transcription of the your CDKN1A gene, okay, which encodes the CDK inhibitor that is known as P21. Clear? So it occurs late in the G1 phase and it is caused by P53 dependent transcription of CDKN1A gene, which encodes CDK inhibitor, that is P21. Now this P21 inhibits CDK4 cyclin complexes, thereby maintaining RB in active hypophosphorylated state and blocking the progression of cells from G1 to S phase, which we have discussed. Okay, this pause in cell cycle is welcome as it gives the cell breathing time to repair its DNA damage. Clear? Now another function of P53, that P53 also helps this process by inducing certain proteins, that is one of these is GAD45. Clear? That is growth, arrest and DNA damage. It enhances DNA repair. Suppose if DNA damage is repaired successfully, the signal responsible for P53 stabilization keys and P53 levels fall, releasing the cell cycle block. Clear? And the cells may then revert to normal state. So this is about cell cycle arrest, in which we have discussed that it started in G1 phase, role of CDKN1A gene, that is forming P21, which is inhibiting your CDK4 cyclin, which is activating your RB. Okay, or also P53 also stimulates another protein, that is GADD45. Okay. Now the second is your P53 induced senescence. So senescence is a state of permanent cell cycle arrest characterized by specific changes in morphology and gene expression that differentiate it from reversible cell cycle arrest. Senescence is the product of epigenetic changes. So senescence is a product of epigenetic changes that results in the formation of heterochromatin at key loci, including genes that are required for progression of cells from G1 to S phase. Clear? So this is about senescence. Now the next one is P53 induced apoptosis. P53 induced apoptosis. So apoptosis of cells with irreversible DNA damage is the ultimate protective mechanism against neoplastic transformation. This is the ultimate protective mechanism. Okay, your P53 directs the transcription of several pro-apoptotic genes such as your BAX, PUMA, okay, and they are believed to tip the balance in favor of cell death via the intrinsic pathway. Clear? Now the next question after these three, that what determines whether a cell repairs its DNA, becomes senescent or undergoes apoptosis is uncertain, but both the duration and the level of P53 activation may be the deciding factors. Clear? Once again I am repeating the question, what determines whether a cell repairs its DNA, okay, or it becomes senescent or it will undergo apoptosis. So this determination is based on your duration and level of P53 activation. This will totally depend on the P53. Okay, its level of P53 and duration of P53 activation. Clear? it appears that the affinity of P53 for its binding site in the promoter and enhancer of DNA repair gene is higher than its affinity for binding site in pro-apoptotic gene. So this is very important point. Okay, P53 has higher affinity in binding to the promoters and enhancer of DNA repair genes. So it is first going for DNA repair, not for apoptosis. Clear? So this is very important point which I have mentioned here. Clear? Now, if P53 is sustained at this level due to ineffective DNA repair or other chronic stress, epigenetic silencing of genes that are needed for cell cycle progression occurs, leading to senescence. Alternatively, if enough P53 accumulates to stimulate the transcription of the pro-apoptotic gene, the cell will die. Clear? Now, irradiation and conventional chemotherapy are the two common modalities of cancer treatment, mediate their effects by inducing DNA damage and subsequent apoptosis. Now, tumors with wild type TP53 alleles are more likely to be killed by such therapy than tumors with mutated TP53 alleles. Okay, such in the case of your testicular carcinomas, testicular teratocarcinoma, childhood acute lymphoblastic leukemia, which usually have wild type TP53 alleles. Okay, so these have wild type TP53 allele. So the irradiation and conventional chemotherapy has good effect on this. Clear? In contrast to these, tumors such as your lung cancer, colorectal cancer, which frequently carry TP53 mutation, are relatively resistant to chemotherapy and irradiation. Clear? A second less obvious but even more nefarious result is that cells with defective P53 acquire a mutated phenotype or tendency to acquire additional mutation at high rate. Clear? Now the last point about P53, that there are certain other family members also, that is P63, P73 along with your P53. Clear? Now this P53 is ubiquitous in nature, so it is ubiquitously expressed. This P63 is essential for the differentiation of stratified, this is essential for stratified squamous epithelia, while this P73 has a strong pro-apoptotic effects after the DNA damage induced by chemotherapeutic agents. Okay, so this is all about your tumor suppressor gene, that is